Hi everyone, this is your instructor Kwame. So this is the last part of our lecture on microbial metabolism. And so we will look at the biochemical pathways that uh, are employed during anabolism. Anabolism basically is the building of larger molecules from smaller units. And so that's how polymers are formed, starch, and cellulose and glycogen. That's anabolism. It requires the use of energy, so that the net energy for anabolic reaction is a negative. There is a net energy loss because more energy is used in forming those bonds than are produced when those bonds are synthesized. So photosynthesis is one such example. And in eukaryotes like plants and protists, you have an organelle known as the chloroplast, which plays a critical role in the process of anabolism. The chloroplast has an outer membrane, and there's an outer membrane, and then it has an inner membrane. In between the outer and inner membrane, there's an intermembrane space. It also has thylakoids. Is a component of the chloroplast where you have pigments, the chlorophyll pigment, which traps the sun's light energy. And then in between the thylakoids, which are stuck together like these, you have the stroma, which lies in between the thylakoids. So if you look at the chloroplasts under the microscope for algae and plant leaves generally, this is how they will appear. Depending on the type of plants, they are arranged differently. In prokaryotes, since prokaryotes do not have membrane-bound organelles, they do not have chloroplast. However, photosynthetic prokaryotes have their inner membrane folded together. So there is an infolding of the inner membrane, and then these inner membranes then contain chlorophyll. So it serves the same purpose as a chloroplast in eukaryotes. So that's become the specialized site for photosynthesis. The production of energy, which is anabolism, which anabolism would depend on, in the process of plants synthesizing their own food using the light rays, or photosynthetic organisms using that, is a two-step process. So there is the photophosphorylation process, which is a kind of one of the ways that ATP is generated, and then there's the process of photosynthesis where the food is synthesized. The two go together. So if you write an equation, we are writing an equation for both processes. Okay. So it's a two-part process. The photophosphorylation process is described as light dependent because there's a conversion of light energy into chemical energy in the form of ATP. And then there is the light independent phase known as the dark reactions, where the chemical energy in ATP is used to convert carbon dioxide to sugar. And that's the one that we are very familiar with, where carbon dioxide is converted to glucose. And that's how plants make their food. That's how photosynthetic organisms synthesize their food. That's the process of photosynthesis proper, this process of converting carbon dioxide into glucose or sugar. And then the first component, which the dark reactions is dependent on, is the photophosphorylation process. So a more closer look at a chloroplast, you have the outer membrane, and that's the inner membrane. And then you have the thylakoids, so these are thylakoids. You have the stroma connecting thylakoids to each other, the lamella connecting the thylakoids to each other. But in between the thylakoids, you have the stroma. So these are the stroma. The dark reactions take place in the stroma. So that's something you should note. the light reactions take place in the thylakoids. If we look at chloroplasts within membranes, how they actually appear is this. They are stuck together and embedded into the membranes, and they are positioned such that they will be able to have direct access to the sun's rays and be able to trap the sun's energy. There are two systems involved in the process of photophosphorylation. There is a photosystem 2 and photosystem 1. Each of them uses different wavelengths. So photosystem 1 is activated when you have wavelength of 700 nanometer of light. So if the light has a wavelength of 700 nanometer, it is photosystem 2 that is activated. So electrons are excited, and with it comes protons as well, and this is then used to generate ATP from ADP. And then this will happen in electron transport chain. And then you have for photosystem 1, you need a wavelength of 680 nanometer for that to be excited, and the electrons are then excited, but then it results in the production of NADPH. Okay. And so ATP for photosystem 2, NADPH for photosystem 1. But these are all taking place in the electron transport chain. And they use the process of chemiosmosis. It's a very similar process. You have light energy hitting photosystem 2, electrons are released, and protons as well. So a gradient is set up, and the balance can only be established when it goes through ATP synthesis. And in the process, you have ATP produced and ADP being a byproduct. For the synthesis of NADPH, it's a different process. It goes through photosystem 1, and you have NADPH produced in the process. But what you need to concern yourself with, especially with this, is that there's an example of chemiosmosis. So there is a proton and electrochemical gradient that is also set up, and that ATP is only synthesized when the ATP synthetase is activated based on the movement of the protons and the energy that is generated. For light reactions, you need photophosphorylation. So for eukaryotes, they have chloroplast. For prokaryotes like cyanobacteria, there's an infolding. There's an infolding of the inner membrane that serves as the chloroplast. So if we do an accounting of the reactants and products, for the light reactions, you need light, you need water, you need NADP plus. For the product, you need oxygen, NADPH, and ATP. Light reactions occur in the thylakoids. Remember, that's where the chlorophyll is. And the purpose of light reactions is producing energy using the electron transport chain and chlorophyll. Okay. And then for the anabolic components of the reactions, which is actually photosynthesis, the purpose is to produce food. And that occurs in the stroma. You need NADPH and ATP and CO2 to produce NADP plus and glucose. And you would have ADP as well as the products, one of the products. So anabolism, an effect in terms of the dark reaction, results in the production of glucose. It occurs in a process also called the Calvin cycle, and then when this glucose is produced, it becomes the source of energy for food chains and the different ecosystems that we have on Earth. So that ATP and NADPH becomes the source of energy, and it is then transferred. Once it becomes part of the chemical component of the food, the plants or these photosynthetic microorganisms are fed on, and the chain goes on, and energy is transmitted through the system. Okay. For the products, for the dark reactions, you have glucose and NADP plus that are regenerated over and over. There are other anabolic reactions. So glycogen is an example of a macromolecule that is produced by a combination of several glucose molecules. When you have glycerol and 3 fatty acids combining, you have triglycerides. That's also an example of anabolic reaction. You have several amino acids that are combined together, you have a protein. Now, these all require the use of energy. Okay, so energy is taken in, more energy is taken in than it is produced. So that's why it's an anabolic process. By way of review, catabolic process yields energy. Anabolic processes end up creating biosynthetic molecules. Okay. So heat is produced in catabolic process, and then utilizable energy is also synthesized in the form of ATP. We have byproducts that are produced that has to be eliminated from the cells, otherwise the cells will die. For anabolic processes, you have external nutrients that are incorporated. You have intracellular precursors that exist in the cell that helps in forming the intermediates and the polymers that the cells need. So if we look at the entire process in terms of catabolism, you have glucose. It's broken down in the process of glycolysis in most organisms. You end up with 2-pyruvic acid and 2-acetyl-CoA, and CO2 is released. And this all happens in the cytoplasm. Okay. And 2-ATP is released or produced in glycolysis. That's the net energy produced. And then you have Krebs cycle, where you have 4 carbon dioxide molecules that are produced. You also have 2-ATP that is produced. The pyruvic acid may also go directly into the electron transport chain after the electrons and the hydrogens in NADH and FADH have been synthesized. Okay, so they can be passed directly on. For some organisms, right from glycolysis, the end products can go directly into the electron transport chain as well. In cellular respiration, you will need Krebs cycle to come in, and the products could also go directly into electron transport chain. Now, in the electron transport chain, you have 34 ATP that are produced. So that's where the most ATP production takes place. Note that you have glycolysis taking place in the cytoplasm. You have the intermediate or preparatory step after glycolysis also taking place in the cytoplasm. You have Krebs cycle and electron transport chain taking place in the mitochondria. In terms of accounting for the input and the output, just another go at it. And you will notice that for the preparatory step, there is no ATP produced. However, you see the other components. You have pyruvate, NAD+, and coenzyme A, which end up becoming acetyl coenzyme A that will enter the Krebs cycle. So it's important that you note the reactants, the products, the purpose, and where all these different components of the catabolic and anabolic processes occur. Okay, it's important that you know. And then the net energy that are produced at each of these steps and stages in the processes as well. So if we compare aerobic organisms, anaerobic, and organisms that carry out fermentation, these are some of the things that we see. For aerobic respiration, oxygen is required. Anaerobic doesn't need oxygen. Fermentation doesn't need oxygen. The type of phosphorylation For aerobic respiration, you have substrate level phosphorylation and oxidative phosphorylation that occurs during the Krebs cycle. And then for anaerobic, you also have substrate and oxidative. For fermentation, you only have substrate level phosphorylation. You do not have oxidative phosphorylation in fermentation. The final electron or hydrogen acceptor is oxygen for aerobic respiration. For anaerobic respiration, it could be some of these inorganic compounds, so nitrates, sulfates, carbonates, or it could be an outside organic molecule. And then for fermentation, it is a molecule that is within the organism, within the system. And in most cases, you have pyruvate being the final electron acceptor. Then the potential molecules of ATP that are produced for aerobic respiration 36 to 38, whether it's prokaryotes or eukaryotes, that's where you have the differences. For anaerobic respiration, it's just 2 to 36, not quite efficient as aerobic respiration. And then for fermentation, you have 2 ATP. And these 2 ATP are just the ATP that are produced during glycolysis. For the anabolic processes, you have the light reactions that occurs in the thylakoids, resulting in the production of oxygen and ATP, as well as NADPH. ATP and NADPH, energy that is stored in these molecules are then used in the dark phase, which occurs in the stroma, and it's used then to synthesize sugar. The food that is then synthesized in the process, you have carbon dioxide that is also used in that process. So carbon dioxide is used to synthesize the sugar. Water is used to synthesize this oxygen in light phase. And then the products for the dark reactions, you have NADP plus and ADP, as well as phosphates. And so it's recycled over and over again. So that brings us to the end of our lecture for microbial metabolism. Now it's a lot of information, so spend some time on both the lecture and the course material, as well as the PowerPoints that I will upload in the form of PDF for you.